Hi, everyone. Dr. Rushing here to give you an OCNDS research roadmap update. So I wanted to start by showing that the number of publications mentioning OCNDS continues to rise from the very first paper back in 2016, which identified the first 5 individuals that were diagnosed with Oker-Chung neurodevelopmental syndrome, all the way to this year in 2024. We're now at 29 publications. And the recent publication that just came out, we will be providing a research explained shortly. But this is really exciting, and we expect this number to keep rising. Next, I wanted to show the blank research roadmap that I presented last year in December and review the main portions of it so that we can dive a little bit deeper into the detail about the updates that we have for this year. So on the top, you'll see a large arrow that says natural history, and this is indicative that natural history is continually collected over time. It's really not just one step in the process, but we continue to do it at each step, and it informs next steps as we go back through the roadmap. And then on the left here, we have 2 major players, the disease models as well as basic science studies. And these really work hand in hand to inform preclinical research, which is really before things go to a clinic. Once we have preclinical research, this will inform phase 1, 2, and 3 human studies, also called clinical trials. And then once these are complete, we also have long-term monitoring efforts that then will feed us back even further to answer new questions that might arise. So when I presented this last, I had details under each of these aspects of the research roadmap, and now I'm going to dive into each one of them on the next slides to give you some updates about these initiatives. So first, starting with natural history, we have our natural history study with Simon Searchlight. And we did a campaign this past February called Shine Your Searchlight. And the goal of this was to increase participation. We had increases in registration of 8 people. So we're now at a total of 126 people registered. 9 people submitted their lab report confirming their genetic test diagnosis and an Oker-Chung diagnosis. We've had 5 people complete their medical history intake form. 8 people complete additional surveys that are loaded into Simon Searchlight, and 1 person submitted a blood sample. So this is really exciting progress. We hope it continues to grow. Another exciting news, we have a new intern from Vanderbilt, Elena, that is helping me to analyze the data that's currently existing in the natural history study. And the goal of working with Elena is to find new insights about OCNDS and publish them so that you all have access to this information. Next, we also have an EEG pilot. So EEG is what measures the electrical activity in the brain. And Dr. Barnett at UT Southwestern has already analyzed 3 sets of patient records. And she's submitting an institutional review board application. So this is a regulatory application so that she can formally analyze more records with the goal of analyzing 20 individuals who have OCNDS. So if you are interested in being part of this study, please do email me so I can get you the right information and get you connected. My email is right here underneath this column. The foundation has also been conducting surveys over the past few years, and we recently conducted a 2024 community survey that received 60 responses. So thank you to those of you who spent the time to fill that out. I wanted to inform everyone of the top reported symptoms, and those were speech delay, global developmental delay, gross motor delay along with fine motor delay, and then short stature. Additionally, the top reported concerns for the current state are education, self-care, hygiene, social interactions and speech were tied for 3rd, and then behavior. Furthermore, the top information requests were for medical research updates, so something that we're doing right now, treatment options, and then sharing of more personal stories from the community. And then lastly, under the natural history umbrella, looking at a disease concept model. So just a reminder that this is a more formal framework to really assess and describe the lived experience of individuals with OCNDS and their families. And this will provide a basis for us to generate clinical outcome measures that can be used in studies, but making sure that they're meaningful to people who have OCNDS and their families. So we are currently on a waiting list with our collaborator Combined Brain to receive an intern to help us conduct a formal disease concept model in fall of 2025. So we still have a ways to go, but we're really excited to be on this list and be receiving help towards this endeavor. Next on our roadmap were our disease models. So I'm going to give you an update about each of the models that we have. First up is our mouse models. They are all in Dr. Rebholz's lab in France. And for the K198R mouse, which as a reminder, K198R is the most prevalent mutation that we've seen in CSNK2A1 that is causative for OCNDS. She has submitted the publication characterizing this mouse, and it's under review for publication. We're hoping that it will be out for the community very soon. The other two lines are not as far along, but the R312W has experiments ongoing to match the experiments that were done with the K198R line. And the R47G mouse has safely arrived to Dr. Rebholz's lab and has finally passed the required quarantine requirements to make sure that it doesn't get other animals sick as it enters the lab. Next up are induced pluripotent stem cell lines. You may remember from December, we have 9 different patient lines stored with Simon's Searchlight. We are waiting for different control lines to be made right now to accompany those patient lines. And 3 of those lines are expected to be completed by the end of this month. And then once those are completed, Dr. Quintelman at TGen will start looking to do experiments to characterize these lines. We do still have our patient-derived fibroblast lines that Dr. Rangasamy at TGen has been working on, and he has been working on data on mitochondrial function. So you might remember back from high school biology, mitochondria being the powerhouse of the cell. So what he has actually found is there's indications of mitochondrial dysfunction, meaning they're not working as they should. So he's collaborating with Dr. Rebholz to do experiments that match his in the fibroblast lines, but with the mouse model, the K198R. And the goal is to have a collaborative publication as soon as possible, hopefully by the end of this year, so that other researchers are aware of this phenotype that they're observing with the mitochondria, and others can do experiments to match. In related news to mitochondria, I will be attending a mitochondrial medicine conference in June to learn more about mitochondria, as well as see if there are any ongoing treatment efforts for people who have mitochondrial disease that might be relevant to OCNDS. Our fly model is currently in use in the Chow lab, and I'm going to provide a study update very shortly in the preclinical research update section. And lastly, with our FITCH model, we do have preliminary results that are on a website called bioRxiv, where early drafts of publications can be put out so that the data is readily available. So Dr. Rangasamy has provided preliminary data that symptoms can be rescued in the OCNDS zebrafish model by supplying normal copies of the CSNK2A1 gene to fish that have the mutations. So this is really exciting preliminary data that we hope can be replicated across other models. Next up in our roadmap is basic science experiments and approaches to OCNDS research. The first update I'm going to give you is about genotype-phenotype correlations. And this is just a fancy term to describe how specific changes in the DNA appear in people as far as symptoms, severity, what will happen. And the new publication that just came out is actually a paper describing new implications for genotype to phenotype comparisons. And this is the paper that the Research Explained will be published shortly for. And what they found was that individuals that have variants in CSNK2A1 that are called null variants, or changes in the DNA that do not actually result in a gene product being made, and thus the protein isn't made, they might have lesser or a diminished amount of symptoms as it relates to things like language deficits, the changes in the facial features, or intellectual disability, and thus have a milder phenotype as compared to individuals who have variants that are called missense variants, or one-letter changes that still typically lead to a gene product and protein product being made. And I'm going to review the types of variants later on in this talk, but this is pretty exciting to have some type of added genotype-phenotype correlation. Next, looking at the mechanism of action and the targets of CK2 when we look at people who have OCNDS. We don't have any new research just yet, but I did want to review briefly the foundation-funded research because they suggest potentially different mechanisms that are contributing. And the first paper here I have in this figure is by Isabel Dominguez, and she used the fibroblast lines. And what she found here, I've used little shapes and colors to try and help simplify this. But under normal conditions, casein kinase 2 proteins will hit or target all of these different other proteins of different shapes, sizes, etc., to do its normal day-to-day function. However, in OCNDS, what they found in this paper using the fibroblasts was that less targets were being hit, and the intensity of that effect was dependent on the mutation. So this suggests that casein kinase 2 loses function or is a loss of function mechanism. Our other collaborators in Germany, which is the second paper here, used E. coli, which is a bacteria, as the model. And I've lowered the number of shapes here just for simplicity to show you the difference. But under normal conditions, casein kinase 2 had these targets. And in OCNDS, what they found was that instead of a reduction in the amount of targets, they found that the targets, or what CK2 acted on, was completely different. So it was a change in the substrates. So this suggests that it's not a loss of function necessarily, but could just be a change in what CK2 chooses to act on. So there's more work that really needs to be done to distinguish between these potential mechanisms and see if it might be different for different variants in the CSNK2A1 gene. And then lastly, under the basic science efforts, one of the big categories we had talked about were biomarkers or biological markers. So ongoing efforts that we did just mention on the other slide are the EEG analysis from the medical records to see if there's anything that stands out in the EEGs that can tell us more about OCNDS. Assays with the K198R mouse model are still ongoing, and in the future, there will be assays that use the patient-derived induced pluripotent stem cells. Moving on to preclinical research, we discussed in December 3 main avenues, and I'm going to touch on 2. The first being gene therapy or gene modification strategies. So we asked back in December, is there any evidence that we can alter symptoms in OCNDS, and maybe there might be a specific window of opportunity for treatment to prevent symptoms or mitigate those symptoms? And so Dr. Rangasami's work with the zebrafish I mentioned on a former slide, we do have preliminary evidence of rescuing symptoms in the zebrafish model by providing an excess of the normal or wild type CSNK2A1 gene. However, additional experiments are still needed to confirm the validity of this effect, and we do need to do it across other models such as the cell lines and the mouse to make sure what we're seeing is accurate. But this is still really exciting preliminary data to suggest that rescue can be an option. The other main avenue we discussed back in December was drug repurposing. And you can think of this as screening with drugs that are already on your pharmacy shelf that are being used for other things, but repurposing them to be used for OCNDS. And so really exciting in the Chow lab, the fly model, the screen is currently 75% complete. And we have 15 different hits of compounds that have improved the OCNDS flies phenotype or their symptoms. And so now we have a compiled list of compounds or drugs that we can now use to test across other OCNDS models and potentially test in patients more quickly to help improve various symptoms. And we will have more details in the end of Q2 on this screen. We are still working with Unravel, our biotech partner, to use their artificial intelligence platform to also find compounds. We are still awaiting sequencing results from the mouse model tissues provided by Dr. Rebholz to complete analysis and screening to get a compiled list of compounds. And there's also a potential future collaboration using human samples. So this work is really still ongoing. And so I'm going to jump to the long-term monitoring portion of the roadmap simply because we do not have any clinical trials or clinical studies ongoing in OCNDS, but we still have long-term monitoring efforts going that feed back to these other parts of the roadmap. One of those is Trend. So you may remember back from the roadmap presentation in December, Trend uses data from the private caregiver Facebook group to generate new insights about medications, symptoms, and so forth. And we expect to gather some refined insights by the end of April, as that will hit our 6-month mark after starting to work with them this past October. So stay tuned for updates on that. Citizen, we will be launching recruitment efforts with them in July. We are very, very excited to be on the lookout for an announcement and how to participate. And Citizen will help us with medical record collection so that you can easily collect all of your child's medical records in one place. And then if you choose to share them with the foundation, so we have ready access to look at the records and see what might be similar across people that have OCNDS. An ICD-10 code. So this is the code that is used in electronic health records or medical records to indicate specific diagnoses. We are partnering with a law firm, Hogan Lovells, to submit an application for the fall meeting to hopefully be approved for an ICD-10 code. This law firm has worked successfully with other patient advocacy groups in the past to get codes, so we are feeling very hopeful that we will have a code. And this will help not only with research efforts, but to identify more people that have OCNDS, and of course, help with treatment monitoring and treatment approvals in the future. And then for long-term monitoring, the other portion of it was clinical guidelines. So we can start to generate these guidelines once 100 people have completed the full medical history intake in Simon Searchlight. Currently, we're only at 79 individuals, so we are not far off. However, it may take closer to 200 individuals to actually generate the guidelines simply because of how different people are with OCNDS and the different variants that we've been observing. So with that being said, a couple of reminders. So please check your Simon Searchlight dashboard to ensure you have filled out that medical history portion. And if you're not yet in Simon Searchlight, please reach out to me so I can help you or connect you with someone that can help you walk through. It's really important so that we can start generating some guidelines. Additionally, even though we don't have any clinical trials ongoing for OCNDS, there are many studies ongoing that could benefit the OCNDS community as a whole and other neurodevelopmental disorders. And we are now sending out monthly summaries to the community that have information about ongoing research studies. Of course, if you have questions or concerns about any of those studies, please feel free to reach out to me directly. And so with that, I did want to provide a brief registry update about the types of variants that we're seeing in the CSNK2A1 gene in our registry. And just a reminder, a variant is a change in the letters that make up the DNA. This is often interchangeable with the word mutation. And this example has been previously used by Dr. Oker in a talk, so I figured this was a good one to use. And for our example, we're going to use this sentence, "The one big fly had one red eye." And this is going to be considered our normal gene. You might often hear this referred to as a wild type gene. And so the different types of mutations that can happen are listed under here on the next lines. And one of them is called a missense mutation. So I'm going to walk you through this. Missense mutations are one-letter changes. And so in the example provided here, the E in the word "the" has been changed to Q. And as you can see, that makes the sentence not really make sense because that's not an actual word. Some examples that you might see in OCNDS are the K198R mutation or the R47Q. And most of the people registered with us have missense mutations. So we have 178 individuals registered with missense variants. The next one that we see are called nonsense variants. And this is what happens in our example when the sentence ends too early. So in this specific example, we see the one big and then the sentence ends. So this is often quite confusing. Some of the annotations you might see in a genetic report are having an X at the end for the letter. And we have 13 individuals currently registered that have nonsense variants. The next one is called a frame shift, and this can be a very confusing one because it can be letters inserted or deleted. And what this does is it shifts the DNA out of alignment, which results in a sort of random sequence that doesn't make sense. So in the example here on the third row, down from the main sentence, you can see that instead of the B in big, it's been replaced with a Q, but the B isn't deleted. So now our words are all jumbled as we go. And similarly, the representation of that on genetic report sort of looks like a jumble of alphabet soup as well here. We have 8 individuals currently registered with frame shift variants in the registry. And then lastly, we have deletions. So deletions can be letters deleted of varying sizes or numbers. There are 3 different example lines here for deletions. So one of them is when just the word fly has been removed, the one big had a one red eye, so we're left confused as big what. You might see it annotated as such with DEL at the end to indicate deletion. Another type of deletion might be a larger deletion where we're actually missing the entire first part of the sentence. And it might be annotated as such where it says exons 2 through 4 are deleted. Lastly, the entire gene can be deleted. So in this in this example, we don't see anything for our sentence, and it's going to look again like kind of like a number and alphabet soup here. And in our registry, we have 23 individuals registered with all different types of deletions. And so you might be wondering, okay, well, what does this mean? Why is it important to know my variant? Why is it important for the foundation to know my variant? So just to touch on this, it really does help with research as well as in the future, it will help with clinical trials. So you're really playing a vital role in accelerating scientific discoveries and helping to uncover new treatment options, not only for you and your family, but for others affected by OCNDS. So if you haven't yet provided your variant to the foundation, just be on the lookout because likely myself or someone else from our team will be reaching out to try and get that information from you. And if you're having trouble obtaining that information, not a problem. We will do our best to try and help you to get that information. Secondly, tailored treatment approaches. So think of the term personalized medicine. Just as no 2 individuals on the planet in general are exactly alike, the same holds true for genetic mutations. So understanding the specific genetic variant that's associated with OCNDS enables you and your healthcare professionals to tailor treatment approaches in the future to address your unique needs. And some people might be doubtful of this, but if you think back to that mechanism slide that I talked about with the foundation-funded research, it's quite possible that the different variants in the CSNK2A1 gene have different mechanisms and may need different approaches. So knowing your variant may be important for tailoring how your child is treated in the future. Predictive insight. So knowing the genetic mutation could provide very valuable knowledge about what might happen over time, helping you and your healthcare providers anticipate the progression of the disease and then potentially, plan for potential complications that might happen. So that one new publication I highlighted suggested that missense variants had a more severe phenotype. So if you know this, you might be able to better prepare or better understand the possibilities of what symptoms might happen. And then lastly, for family planning. So the genetic mutation not only impacts you or your child in this case, but it can also have implications for your family members, or if you're planning to have more family members. By identifying the gene variant, you're going to gain really valuable information to help you with family planning decisions, and you could benefit from genetic counseling services to better understand how it's inherited and then, of course, potential risk to future generations. So I hope this helped you a little bit to better understand why having the genetic variant information is really important, not only for you, but for the foundation and the research efforts. So with that, I will take any questions, and I hope this helped you better understand where we're at with our research roadmap. Thank you so much.